Hello, my name is Mayra. I'm from Mexico and I am in the first stage of the PhD in Ciencias del Mar y Limnología. And I am working on the effects of microplastic on the demography of freshwater microcrustaceans. In this talk, I present the results of our experiments on the effects of temperature on the acute toxicity of Daphnia pulex. In the last 10 years, the study of the effect and concentrations of multiple stressors have special relevance and has been a great concern for the scientific community. The ecosystems are simultaneously exposed to complex mixtures of contaminants and to considerable variation of abiotic factors such as temperature, wastewater discharge, and pollution by plastics. Since 1950, the production of plastics has increased almost 200-fold. In addition, it's common that some water bodies are used as industrial, pharmaceutical, and domestic wastewater receptors, which, in addition to containing chemical pollutants, could have a high content of plastic residues and personal care products which reach aquatic ecosystems. There are two main types of microplastics, primary and secondary. The primary are plastics produced industrially as raw material for some other products, mainly cosmetics. And the secondary are the smaller plastics derived from the fracture of macroplastics due to dragging and erosion suffered by being exposed to high temperatures, prolonged time in aqueous media, or from physical damage. Usually, the microplastics are divided in five categories based on their size: megaplastics, macroplastics, mesoplastics, microplastics, and nanoplastics. Microplastics are usually mostly fibers, fragments, and spheres from personal care products such as scrubs and toothpaste. At present, there are more than 5,000 types of synthetic polymers, which makes their analysis complicated. The plastics most used in the world are polyethylene, polypropylene, polyestyrene, polyvinyl chloride, and polyethylene terephthalate. The effect of plastics have been studied in the ocean. Enormous quantities of plastic has been recorded in the form of islands in the Pacific Ocean and in the stomach contents of fish and birds due to the direct ingestion. However, in epicontinental waters, there is still no record of its presence and the effect on the organisms with which it has contact, such as aquatic zooplankton communities. Rotifers and cladocerans are generally filter feeders, capable of consuming particles between 20 to 100 micrometers, which allows small particles of microplastic between 0.05 to 50 micrometers to be easily ingested, and this could have an adverse effect on their survival. In addition to their importance in the food web, it has been documented that many species of zooplankton have been used as bioassays organisms in acute or chronic toxicology, or as bioindicators of contamination, because they have special characteristics such as sensitivity, high rate of population growth, and many are cosmopolitans. Also, some works shown bioaccumulation and therefore could transfer products in tissues of top predators such as fish. The composition of plastic fragments allows their persistence in the aquatic environment, in suspension, and in sediments. It is even known that they are constantly under degradation processes that promote the release of toxic chemical substances. The temperature is a determining factor that affects the composition, and it is known that conditions such as staying in aqueous or wet environment and an increase in temperature play an important role in the degradation of the plastics. Temperature also plays a very important role in the life cycle of organisms. It can affect variables such as average lifespan, reproduction, feeding, biochemical processes, and metabolic rates. The effect of microplastics on aquatic organisms can be diverse, from intoxication associated with chemicals to mechanical damage by direct ingestion of fragments or spheres. Carolyn Burns in 1968 conducted a study of ingestion with 7 species of cladoceran and microbeads of plastic with different size. In this study, we can see a relationship between body size of cladocerans and the bead size ingested. From 293 individuals tested, 175 ingested plastic beads. Hofscher in this year studied the effect of ingestion rates of microplastic beads by Daphnia magna and Daphnia pulex with different temperatures. We can observe the increase in the ingestion rate of microplastic beads in conditions of low food and high temperatures. The information of concentration, persistence, and type of microplastics in freshwater systems is limited. For this reason, the effect that this may have on population of invertebrates such as zooplankton have not been exhaustively studied, even knowing that some species may have adverse effects that affect other groups, either due to bioaccumulation or not being available in the system. Publications related to this topic began in 2010, with less than 5 articles per year. Currently, that number has increased exponentially. According to the Biological Abstract Database, there are more than 1,000 articles related to the keyword microplastics, of which only 4% refer to studies in epicontinental environments. Cladocerans with considerable size difference between neonates and adults are common in epicontinental waters, yet there are very few studies on the consumption rates of microplastic by neonates, juvenile, and adult cladocerans. Here, we present the effect of temperature on the consumption of microplastic by different life stages of Daphnia pulex. First of all, we select the size of the microplastics and vote on Sigma Aldrich Labs. For the experimental design, we select individuals from different life stages of Daphnia pulex, neonates, juveniles, and adults, and 5 microplastic concentration, 2.5, 5, 10, 20, and 40 milligrams per liter, with a control group and 4 replicates for each treatment. We place 10 individuals of each stage in borosilicate glasses with 10 milliliters of distilled water and the microplastic concentration with Chlorella vulgaris as food. The same experiment was set up for 20 and for 25 centigrades. After 24 hours, the surviving organisms were counted and the percentage of mortality was analyzed for the 3 stages of the cladocerans. The acute toxicity test of Daphnia pulex did not show an evident adverse effect at 24 hours under the tested concentration in any of the stages for which the LC50 could not be calculated. We found that the consumption of microplastic and its adverse effect was greater at 25 than at 20 centigrades. The adverse effect of the microplastic was more lethal on the adults than neonates and the juveniles. Eventually, the adverse effects of these substances will be evident on the entire population. With regard to temperature, it can be observed that it is a parameter that greatly influences the feeding behavior of this species because an increase in temperature accelerates the metabolism of the organism, causing an increase in the filtration rates. An increase in the filtration rates increase the probability of encountering with microplastic particles, and it could lead to an adverse effect on survival. In conclusion, we observe that the organism at any growth stage of Daphnia pulex is able to consume microplastics and store them. The adults show a higher sensitivity to microplastic in compared with the juveniles or neonates. The neonates show least sensitivity to the effect of microplastic in compared to the other growth stage, and an adverse effect of microplastic on all developmental stage of Daphnia pulex was more evident at 25 than 20 centigrades. We thank to UNAM, PhD program Ciencias del Mar y Limnología, FESIS Tacala, CONACYT for the scholarship and the project PAPIT. I hope you enjoyed the talk and thank you very much for your attention.